All right, this is photosynthesis part 2. We've covered at this point up to the light dependent reaction. So now we're going to pick up here with step 2, which is the light independent reaction. Now this takes place in the stroma of the chlorophyll molecule, or of the chloroplast molecule. This also is referred to as carbon fixation because it's going to take carbon dioxide, a molecule that's an inorganic molecule that we can't use, and fix it into a glucose molecule, which is a molecule that we can use. But to go from carbon dioxide to glucose, we have to go through a series of steps or reactions that are called the Calvin cycle. And in the Calvin cycle, you take carbon dioxide. There's an enzyme right here at the beginning of that called ribisco. That'll be key later on. That will bind carbon dioxide into this cycle and start building glucose. Now this cycle's got to spin 6 times to build an entire glucose molecule because it only does 1 carbon dioxide at a time. But it will do 6 to build the glucose molecule. But this is a wheel that has to turn, and any wheel that has to turn, you first have to add a little bit of energy to get it to turn. So the Calvin cycle uses ATP energy. Now if you remember from the first step of photosynthesis, it made ATPs and NADPH molecules. Here is the ATPs being used. It's going to take 12 ATPs to make the glucose. Okay? So it gives it a little bit of energy to turn that wheel, and as that wheel turns, now it's ready to build the molecule. Well, what does a molecule need to be able to make bonds? It needs electrons. So where does it get those electrons? Well, here come the dump trucks. They're rolling in with electrons, and they start dumping electrons now into this process. They're going to dump electrons into building this glucose molecule. So carbon dioxides are being attached together with the addition of these electrons and the energy from ATPs that was made earlier. Now, you will get eventually what we call a G3P molecule, which is a 3-carbon molecule that will be released, and then those 2 3-carbon molecules will be put together to make a glucose. You also have to add a little bit more energy over on this side to keep that cycle going so that it continues to make these molecules that we need. The problem here is that this creates a ATP debt. There's more ATPs needed in the Calvin cycle than what is made in the light dependent reaction. Well, in any biological system, that would be something that wouldn't evolutionarily evolve unless there's a solution. The photosynthesis has a solution. It uses what we call cyclic electron flow. So the Calvin cycle needs more ATPs than it is made. So more than what photosynthesis can make is what it needs. So I've redrawn here the thylakoid. And you've got the electron transport chain that makes ATPs. If that just goes through normal processes, it doesn't make enough ATPs down here. Cyclic electron flow, it allows this electron here, that instead of jumping out here and being picked up by NADPH, it will allow it to cycle back to this protein and go through this proton pump again, repeatedly, over and over and over. And what that does is that continues to give energy to this pump so that it will continue to pump hydrogens in here, creating a larger hydrogen concentration gradient than what would be made without cyclic electron flow. And that then generates more ATPs, enough ATPs to power the Calvin cycle. So nature has a way of taking care of itself there. The next thing I want to look at is that important molecule ribisco. Ribisco is the enzyme that binds carbon dioxide into the Calvin cycle. However, when ribisco binds carbon dioxide, it does so when carbon dioxide levels are high. If carbon dioxide levels are low, then ribisco will bind oxygen in a process called photorespiration. That is a negative for the plant. It doesn't do any good for the plant. It uses up energy and takes up a process that gives no end result that the plant can use. So this is not good. Whenever temperature is high, what we notice is that carbon dioxide levels are low, which means photorespiration is high. I've demonstrated that here in a couple of graphs. So as temperature increases, the level of carbon dioxide decreases, an inverse relationship. Over here, as carbon dioxide levels increase, okay, photorespiration decreases because less photorespiration is taking place when there's a lot of carbon dioxide. All right, so now let's see what plants have evolved to deal with those high pressure or high temperature environments. So in a normal cell, you have a C3 pathway. You take carbon dioxide into a mesophyll cell, one cell, does the Calvin cycle, produces glucose. This is plants like rice, wheat, soybean, those types of plants. An evolutionary process that's evolved to deal with photorespiration is C4. In a C4 pathway, you have two cells involved, and the stomata can partially close at different times of the day to try to keep carbon dioxide levels high in the cell. So they will take in carbon dioxide into the mesophyll cell, turn it into malate, which is a 4-carbon molecule, thus the name C4, and store that until it's needed. As it's needed, malate will go into a bundle sheath cell, the other cell, and break down into carbon dioxide, thus keeping carbon dioxide levels high in the bundle sheath cell, and pyruvate. Those carbon dioxides will then go through the Calvin cycle and make glucose. And we see this in organisms like sugarcane, corn, and grass. The next pathway is a CAM pathway. Now this one also only uses one cell, but the stomata are able to open at night when carbon dioxide levels are higher and close during the day when temperature is high and carbon dioxide is low. So at night, carbon dioxide comes into the mesophyll cell. It turns carbon dioxide into an organic acid called CAM. That is stored in the central vacuole. And then whenever it's needed in the day, it will break that down into carbon dioxide that will enter the Calvin cycle and make glucose. And this is seen in pineapples, succulents, and cacti. This is, once again, stomata open at night, allowing carbon dioxide to come in, close during the day, and they just break down this organic acid to make carbon dioxide to be used in the Calvin cycle.